Okay, well, thank you, Andrew. That was a really fantastic introduction. Set me up for explaining what I've been doing with the data. So, as Andrew said, the human genome is a very large object. It's 3.2 billion bases long, and it's incredibly expensive to measure all of those positions in our genome in all individuals. That would be vastly expensive. And so the kind of standard approach in the field is to try to pick a subset of those positions. And the art really is to pick those positions very carefully, measure those positions, and then we can infer a lot, we can learn a lot about our genetics from that. And so the approach that Biobank took was to measure 830,000 positions, and they did that in collaboration with a company called Affymetrix, who designed a genotyping assay called an axiom array, and this was run on all 500,000 people. And what this slide shows is how those 830,000 positions were selected. And so a committee was brought together to try and pick these positions. A large chunk of the positions on this set were chosen somewhat to uniformly spread throughout the genome. There were a large number of positions in our exome, which are the coding part of our genome. And then there were lots of positions that were known to be important to lots of different diseases, and then positions that were important to response to drugs. And so this chip was run on 500,000 people, and because this data is of great interest to many researchers, we tried to do a lot of work up front for the researchers. So quality control and various computational processes were run on the data to avoid every single researcher repeating that process. And we aimed to provide lots of auxiliary information about the ancestry we could derive from the genetic material, about close relatives that we were able to uncover in the data, and also provide the data in a form that researchers could easily use. So one of the most interesting things that we've been looking at from the genetic data is to look at the ancestry that we can uncover. So people's self-reported ancestry, but we can also look at that in a completely agnostic way from the genetic data. What this plot shows, each point on this plot is an individual. They're colored according to their self-reported ancestry here on the right. And we've grouped people broadly into different continental groups. And so you can see that there's a large number of people who cluster as European, a large number of people who cluster as African, and a large number of people who cluster as East Asian. But there's a large spread of individuals between that. And that's really is showing that Biobank has a very large and diverse set of ancestries. One interesting thing that's under development at the moment is that we're going to start to take the genetic material from each individual and try to ascribe proportions of their genetic material to different parts of the UK. So, for example, I might take an individual and I might be able to work out that a large chunk of their genetic material has ancestry from North Wales, a large amount has ancestry from North Yorkshire, and a smaller part has ancestry from Lincolnshire. And so we'll be able, in the future, over the coming year, to be able to provide that kind of resolution. We've also been looking at relatedness. So we can take the genetic material of any two individuals and compare them and try to infer how closely they're related. So if they're twins, identical or non-identical, if they're parent offspring or cousins, we can detect that very well. And so here are some of the numbers that we've uncovered. And I think it's quite striking that almost 150,000 people in the UK Biobank have at least one relative or cousin or closer somewhere else in the data set. Another interesting number is there are around 22,000 detectable siblings. And that's actually much larger. It's actually double what you'd expect by chance if you'd just taken a completely random sample from the UK population. And of course, we think that's because it's not a completely random sample. You know, there's an effect, I think, where if you've been in the study, you might tell your family and your close relatives about the study, and if you're positive about it, you might encourage them to join. Of course, it was a more clustered way of collecting data than a completely uniform process. So as I said before, just to recap, after we've filtered and cleaned the data, we ended up at around 800,000 positions we've measured. But as I said before, there are 3.2 billion positions in the genome. So one question we have, and this is reasonably routine in my field, is what can we say about all of those other unmeasured positions just by using this small fraction that we've measured? And so this slide shows a cartoon of the data. So each pair of rows here represents an individual, and each column represents a position in the genome. And here I only have 4 individuals and 7 positions. In reality, there would be 800,000 positions and 500,000 people. But it gives you the idea of the data. At each position, you have 2 letters in each individual. But we also, in the human genetics community, have other genetic databases. So, for example, there are databases from the UK 10K project and the Haplotype Reference Consortium that contain many fewer individuals, but at a much denser set of positions. And so why is that of value? Well, what we can do is we can take the genetic code of the Biobank participants, and we can try to find matches of sequence, so short stretches of sequence, that match with the genetic material in these databases. And because we all have shared ancestry, we have shared genetic material, and we can use that shared genetic material to predict or fill in the gaps in the UK Biobank dataset. So if I find this match between these 2 chromosomes, I can use this dense chromosome to copy down or predict the missing information. And so this is something that's relatively standard and routine in our field, and we understand how accurately it can be done. The accuracy is generally quite high, and it basically means that we can fill in lots of these gaps and massively enhance the genetic data set. So in total, we can go from 800,000 positions to around 96 million. So over 100-fold increase in the amount of genetic data, having not had to go back to the participants at all. This is purely computational, using available genetic data sets. And the power of that, it's like having a microscope. You can zoom in much more detail on any given position on the genome. It allows people who are working with the genetic material, the genetic data, to make more discoveries. So to give you an idea of what one of these studies looks like, this is an image which shows results of what we know about genetics for your height. So height is a very genetic trait. This plot shows the whole of chromosome 2 from start to end. And each of these red dots is a position in the genome we know has an influence on your height. And when the dot is high up, it means it's more important than when it's low down. And so we already know a lot about the genetics of height. So you can take this information and you can say, well, what does Biobank add? What do we learn when we start to use the genetic data? And a real power here is the size of Biobank. So if I take just 10,000 UK participants and I look at all of the variants I've collected on chromosome 2, and each one is represented by a black dot, you can see there's not very much signal. And what I mean by that is there's not very much of these black dots above this red line. If something's above this red line, we start to get excited. And with 10,000, you don't see very much. If we then increase to 150,000, and this was the data set that was released back in 2015, you can start to see some of these black dots are going above the line. And some of them, or many of them, are in the positions that we already knew. Then we can take it a step further. We can go up to 350,000. And now you can see there are regions that we're discovering for the first time. And there are many of those regions. And so we're making novel discoveries of genes that are influencing our height. And then if we add on the imputation, if we now go to 100-fold more positions, it looks like this. And so but going back and forth, you can really see the value of doing this computational prediction. You get much better resolution, you get a much clearer image of which regions are associated. Now, my role has not been to then take these positions and try to understand them, but we wanted to do a little bit of that, just to try to validate whether some of these are really real, and so I've focused in on this one right at the end. You can see there's that peak. So if I really zoom in and look at that, it looks like this. So now you can see that there are these black dots, which are positions of the genome, and they're above this line, telling me that I've potentially made a novel discovery. And these positions here in blue show 3 genes in this region. And so these black dots overlap with these genes, suggesting that these genes are the important ones. When I add in the imputation, not sure if you can see that too well, but there are quite a lot of grey dots on this plot now, and some of them are higher than the black dots, suggesting that I've increased the resolution. I've found more interesting positions. So the question really is this real? Because in these experiments there is a chance, there are various factors that can mean that this would be a false positive. So what we did was we went and asked some collaborators who had a completely independent set of data what they see in this region. And if I do that, I add in these blue dots, and you can see some of these blue dots are now very close to the line. And that's significant, in this context, significant what we call replication. We've repeated and found the same signal in this experiment. And so the expectation is, if I go back to this plot, that this will happen at all of these, or many of these positions. Just to reiterate again, this is just chromosome 2. Yeah, so there's many other chromosomes, and they look just like this. So when this data is released, there is going to be, I think, a flood of novel discoveries along these lines for many, many different traits. So the data is just about to be released. I think it will go live, as it were, in July 2017. The data that we've been working on is compressed, but even then it's very large, 2.1 terabytes of data. And we've worked hard on providing it in formats that people can assay quickly. And so the release mechanism is that the data is being released to researchers right now in an encrypted form, and then someone will shout go, and everyone will be sent these decryption keys. They will decrypt the data, and then they will be able to start working on it. And I think the current count of people who have access in general to Biobank is 5,000. I think many of those people are accessing the genetic data. So people really are starting their engines right now. I think over the summer, lots of people in this field are going to be very excited, making novel discoveries. So it's been a real privilege to be involved in this. It's the most impressive data set I've been involved with, both from the vision and the enormous amount of effort it's taken to collect it. And it's also incredibly, because of the design of the process, it's an incredibly clean data set, incredibly easy to work with from my perspective. As you might imagine, there are lots of people who've been involved in this. I've listed some of their names here. I just want to highlight a few people, especially Claire Bycroft and Colin Freeman, who work in Oxford, who a lot of the results that you saw on these slides were generated by them. Thank you.